Bacteria divide by binary fission. One bacteria divide each 20 minutes can be billions in a day. Production of the human insulin. Let's take a look. Production of recombinant plasmids in general, in genetics, human pancreatic cells. We take the DNA encoding human insulin. By restriction enzyme, we cut this gene and put it in the plasmid of bacteria, which contain ampicillin and tetracycline resistant gene as a marker. Here we put the gene of insulin and DNA ligase, tie the gene, recombinant plasmid is formed, and DNA fragment and joined by themselves. They are called non-recombinant plasmid. Transformation and selection of bacteria. We need to select the plasmid are then mixed with the bacteria E. coli. These introduce the plasmid into the host cells. Only some bacteria actually pick up the plasmid, and only some of them are recombinant. Recombinant plasmid, we need to differentiate between the recombinant plasmid and non-recombinant plasmid, and we put a marker. Firstly, the plasmid used are a carrier-resistant gene for antibiotic ampicillin. To select the transformed bacteria, first culture the bacteria on a agar plate containing the ampicillin. The ampicillin resistant gene in the plasmid, that is transformed bacteria with a recombinant plasmid, will live on this agar. Each colony to select agar plate containing tetracycline. Also, we plate this bacteria on a plate containing tetracycline. The bacteria can, from the result of these two agar plate, we can identify the colony formed by transformed bacteria with the recombinant plasmid and select these bacteria. Colonies are determined exactly. Manufacture of the insulin. We put it in fermenter. In each bacteria chromosome, recombinant plasmid will grow, and the bacteria divide, copy to the daughter cells. As a result, many copies of the DNA encoding human insulin are produced. We use the growth of bacteria as a factory for producing insulin. The polypeptide of the bacteria and the functional human insulin are produced. A nice night, Mark and Emila decide to meet and have dinner in a very good restaurant, prestigious and famous. After they have dinner, they return to their home. But they feel something wrong. She is very plant food contaminated with bacteria. These bacteria grow bacterial growth. Very important topic. Let us take two examples to understand the importance of the bacterial growth. Let us take two examples to understand the importance of the bacterial growth. Insulin production in bacteria. The component of human insulin is mainly produced either in E. coli or Saccharomyces cerevisiae. We make the bacteria as a factory for producing human insulin, and this is from 1992. In food poisoning bacteria enter the GIT and grow exponentially and secrete toxins and cause diarrhea and vomiting. Bacterial growth. We speak about numbers of bacteria, not the size. Number accumulating a colony. Group of cells can be seen by naked eye on the colony. There are requirements for the growth of bacteria. Physical requirements, chemical requirements. Physical requirements, the pH, osmotic pressure, temperature. Chemical requirements, carbon, nitrogen, sulfur, phosphorus, trace elements, oxygen, organic growth factors. The temperature, there are maximum growth temperature, and this is the highest temperature growth is possible, and there is optimal temperature and temperature at which the species grow best, and minimum growth temperature, the lowest temperature growth is possible. There is a range, minimum, maximum, and optimum temperature for every species or every bacteria. Three groups according to the range of temperature. Psychrophiles, cold-loving microbes, grow in refrigerator. And this is the temperature of food spoilage. Microbes, zero optimum temperature, range from 10 to 20. Mesophils, optimum temperature 37, range from 20 to 40. Thermophils, optimum 35 centigrade, range from 30 to 35. The pH requirement. Most bacteria grow in narrow range near neutrality between 6.5 to 7.5. Acidophils below 4. Example, lactobacillus. Alkaliophils from 8.5 to 11.5 according to its nature of metabolism. Aerobes, capable of grow in presence of oxygen. CP, Pseudomonas, Pasteurella. Facultative anaerobes, able to grow either in presence or absence of oxygen. Example, E. coli. Microaerophils grow in oxygen concentration less than air. Obligate anaerobes, smallest amount of oxygen is toxic, like Clostridium species or anaerobes. Aero-tolerant anaerobes can use oxygen but tolerate its presence. Aero-tolerant anaerobes can't use oxygen but tolerate its presence. Bacterial growth methods. Increase in numbers, binary fission or budding, either by binary fission or budding. Binary division, the one cell become 2, and the 2 become 4, and 4 become 8. During cell division, cell membrane elongated, and the two types divide, and cell wall and cell membrane begin to form transverse septum, and transverse septum become completely separated longer cells. And in the phases of bacteria, lag phase, log phase, stationary phase, test phase. This is the life cycle phases of growth. The lag phase adapted to nutrient and log phase, active growth, stationary phase, the death equal to growth rate, death, nutrient consumed, and the pH too low, optimized care in production. And this is the curve, log growth, this is the number, this is the equations. Here we find the time versus the viable organism per mil, and this is the lag phase, exponential, stationary term. Measuring the growth, we can measure the growth of the bacteria by direct count, serial dilution, most probable number, and this is the direct microscope count, the drop Hauser counting chamber, and the cover slide, and it sieves under the cover slip. Suspension fill show space of known volume over grade. Serial dilution is a stepwise dilution of the substance in solution. Usually the dilution factor at each step is constant, resulting in geometric progression of the concentration in logarithmic fashion. And this is serial dilution. We put 9 ml for every tube, and we take from the stock solution 1 ml, and we take 1 ml from this to that, 1 ml from this to that, and 1 ml from this to that, and this is the dilution. Here 1 to 10 dilution from the stock, and this is 1 from 1 to 100 of the stock, and this is 1 to 1000, and this 1 to 10,000, and we cut. Culture these serial dilution in plates, and we count the colonies. This is an example. This 78 colonies, 83 colonies, 81 colonies, and we take the average from these colonies and we multiply it with the final dilution factor, and this is the number of colonies per mil of the culture. This is the final dilution we reach. Measuring growth count, turbidity, we use spectrophotometer, and these obligate aerobe, anaerobe, microaerobic, facultative anaerobe, and we can grow the anaerobe in special just back. called candle jars or jazz bags. We group the in candle jar microaerophilic and in gas bag anaerobe and facultative anaerobes.